All right, welcome to our video on enzymes. So the first thing we're going to start with is a general term, and that term is catalyst. Now, a catalyst is going to be anything that is going to speed up a chemical reaction. It's a general term. You also may come across the word catalyst in history class or in an English class, because a catalyst is anything that's going to get something going. So you might talk about, why did the war start? What was a catalyst for the war? That might be anything that got it going, got it started. However, in science class, catalyst is going to be specific to talking about what makes chemical reactions speed up, go faster, get going. So here's an example. Here's a catalyst that speeds up a chemical reaction. So here is a chemical reaction written right here. We have hydrogen peroxide, H2O2. You may never have seen the formula for this before, but you probably all have hydrogen peroxide at home. It comes in a dark brown bottle, and it's that clear liquid that if you pour it on a cut or something, it's going to make those little white bubbles. Now, hydrogen peroxide is made in cells, and it is a reactant, so it's something that's made in your cells. It's going to come out. But hydrogen peroxide on its own is actually going to be toxic. It's harmful to your cells. So you have a catalyst, a chemical reaction that's going to occur and be sped up, where these reactants, hydrogen peroxide, are going to be turned into water, 2 molecules of water, and 1 molecule of O2. Now, we know H2O is going to be in a liquid form, but oxygen, O2, that's going to be a gas. So that's that bubbles, you might say. And this is your product. So reactants go in, products come out. Now, hydrogen peroxide in small amounts is not harmful. However, if we were to allow hydrogen peroxide to build up in our cells and not have this chemical reaction occur where it's broken down into water and oxygen, which are not harmful to our cells, hydrogen peroxide in large amounts is actually a really, really strong base. I almost said acid. A really strong base. And right here, what you can see on this guy's hand, this is not chalk or powder. These are actually chemical burns from coming in contact with a strong concentration of hydrogen peroxide. Now, granted, the stuff you have at home is very, very weak and wouldn't do this to you, but you still have to be careful with hydrogen peroxide. So in chemical reactions, in order to get from reactants to products, so what we start with and what we end with, we are going to need to break bonds in our reactants and form new bonds in our products. So this hydrogen peroxide, in order to make water and oxygen, we have to break apart these bonds between these hydrogens and oxygens and then reform new ones over here. Now, biological catalysts. So catalyst is just anything that speeds up a chemical reaction. Biological catalysts are called enzymes. And this is what this unit is on. Enzymes are going to be molecules that are found naturally, made in our bodies, that are going to speed up chemical reactions. So here's your next one. So enzymes, they're going to speed up chemical reactions. Now enzymes, like your carbohydrates, remember carbohydrates, you had glucose, sucrose, lactose. They had a nice ending to them, OSE, so you could look at the word and say that's a carbohydrate. Enzymes are also kind of nice that way. They have endings with them. The enzymes are usually going to end with the suffix ase, A-S-E. For example, we have lactase, sucrase, maltase, peptidase, lipase, and nuclease. These are all enzymes, and they're easy to see just from looking at the words that they're enzymes based just on this A-S-E ending. So that's an easy way in context if you're trying to figure out what a molecule is, find that ending. If it's ase, it's an enzyme, and it's going to speed up chemical reactions. All right. So before you can make new bonds, so if we have our reactants, remember these are what goes into a chemical reaction, but once we break those, but before we can make new bonds to form our products, we are going to need energy. So to break those existing bonds, it takes energy before we can form those new ones. The energy needed to break those bonds and make the new ones, that's called activation energy. So right here, this graph right here is something you'll get very familiar with. If we look at our x-axis, we have enzyme catalyst reaction pathway. So it's basically showing time. This is time is moving over here, and energy is over here. So our reactants start right here. So let's imagine if we go back to our hydrogen peroxide example, we're starting with H2O2. That's our reactant, what we started with. Now, in order to turn H2O2 into the products, which are water and oxygen gas, we are going to need to put a little bit of energy in. And if you notice, see how this pink line right here, it peaks up. So that's showing an increase in energy right here before it's able to go back down to form your products. So that energy right there where you see an increase after your reactants, after that Y, that's your activation energy. Now, enzymes work by lowering that activation energy. So that energy that's needed to start a chemical reaction, enzymes actually lower that, so it costs your body less energy to get a reaction going, which in turn speeds up that reaction. So here we have that same graph before where we have energy on our Y and time on our X. Your reactants are over here, sort of at the far left-hand side, and your products are down at the far right-hand side. The pink line is showing you what's happening with an enzyme. So it's showing you that you notice that that peak, the highest point, apex on that line, is lower than your purple line, or blue, I'm not sure what color that is, without the enzyme. That's showing you that that enzyme, this reaction with the enzyme is less energy costly than without the enzyme. So it's going to go faster, requires less energy. So a molecule that is an enzyme, a molecule that the enzyme is changing is called the substrate. So right here, this yellowish blob down at the bottom, that's your enzyme. Enzymes have a special place where the substrates are going to attach. So when the substrate, the thing that is going to be broken apart or built up by your enzyme, that is the substrate. So right here, most enzymes are proteins. There are some enzymes that are RNA-based, but we will not talk about those in this class. So just think of enzymes as being proteins. So right here, this is an image of a protein, and then in white, they have the active site, so that place where your substrate is going to attach in order to be broken apart or put back together by the enzyme. And here's a little better drawing of it. There we go. So the folded shape creates a known active site. It's a special part of an enzyme that binds to the substrate. So again, light blue right here is your enzyme. It has a specifically shaped place where that substrate is going to attach. That surface in that area where the substrate attached is known as the active site because it has to be structurally perfect to be able to fit that substrate right in there. If you change the structure of this active site or this enzyme, it won't fit with the substrate anymore and the enzyme won't work. Now there's a special term for this. When the enzyme and the substrate join together, they're going to form something called the enzyme substrate complex. It's just the term given to that molecule that has the enzyme and the substrate joined together into one structure. Oh. So right here, what I want you guys to see is just thinking about these terms reactant and product in terms of enzymes. So in green right here at the top left-hand corner, starting out, you have your green enzyme and your substrate. These are your reactants. So these are the raw products that are going to go into our chemical reaction. They join together in the next step to form your enzyme substrate complex. Once that substrate is connected to the active site, that enzyme can really get to work lowering the activation energy of that reaction, increasing the speed of it, breaking that molecule apart or building it up, depending on what the enzyme is trying to do. So in this case, this enzyme is going to break that orange enzyme apart into the yellow and the purple parts. And then afterwards, we're going to end up with our product. So we're going to end up with our green enzyme. Notice how it's unchanged. And we end up with our yellow and our purple product. So this is what happens in an enzyme reaction. From the reactants, starting with your raw substrate and your enzyme, all the way to the end where we have our final products and our enzyme still. Now, because enzymes have a specific activation site where the substrate is going to bond, enzymes are going to be specific to only one substrate. This is often referred to as the lock and key model. So just like you may have house keys, you may have, well, you guys probably don't have car keys, but you know what car keys are. So if you tried to put your car key in your house, it's not going to open the door because that car key is perfectly formed to only fit in that car lock. So enzymes are exactly the same way. Enzymes are perfectly formed so that only the one substrate is going to fit in their active site. No other enzymes are going to, no other substrates are going to fit there. So it's called lock and key. They only work on one specific substrate. We're going to pause here and you'll have to come back for part 2.